history, and you can talk with him in a plain language. So well, take advantage of that for students, I mean, and for young researchers is essential. So let me show.
This time he resigned at the conference in Valencia.
Yeah.
Oh, but that's duty. You call. God, cancel the trip to Valencia. There's preference.
So
There are preferences and priorities in your life.
so yeah, he's with us, resigning an invitation to be a main speaker in Valencia in Spain. So thank you very much, Vlad. So jumping into the official part of the story, Dr. Vlad Parpura, Professor Vladimir Parpura, is a distinguished professor and director of the International Translational Neuroscience Research Institute in Zhejiang in China. We show that he saw it coming, this dark time that we are suffering in Argentina and now in the US. He saw it coming and he went to China 3 years. 2.5 years ago. So he saw it coming. So ask him, the number of the lottery for tomorrow, probably he will have the answer. So okay. And his talk is going to be astrocytes, release glutamate, but regulated exocytosis in health and malignancy. Thank you, Vlad, once again.
Oh, well, now I have to live up to this expectation, right? It's a little bit too high, huh? Okay, is it good now? Maybe I don't. Is it good now? Is it good now? Do your magic. I think there's echo. It's bizarre when you hear yourself twice. Can you tell me when I can start? Okay. Oh, they cannot hear me. I can talk like they can. Are we good? Oh, I think we are good. Can you hear me? We are good now. Okay. This is the distance. This is the distance. Okay. I'll keep. It's okay. It's okay. It's okay. Don't worry. It's okay. Don't worry. Okay. Can you guys hear me in the back? Thank you. Okay. So what I would like to do is I would like to present you this story. And the way that Javier put it, I feel like a history relic, but what I would like to do really is to take you through those 30 years since discovery by giving you a blend of published data and unpublished data as we go. Please do stop at any given time and ask questions. Here's what I want to do first. I ask Juana.
¿Para qué llamó los chicos?
But there was an echo for him. Yeah. Oh, so it's a magic. That's okay. Oh, okay. Yeah, it's okay. We continue. Okay. He can. So I'll stay a little bit away. Okay. So what I would like to do is talking to Juana and who will be in audience. I would like to give younger people a little bit of a my career path and how did I get to China? What Javier mentioned. So what is this? A dog. Okay, well, my grandma would know that, but which kind of dog? Dalmatian. Very good. What is this? It's an egg. Dalmatian egg. Okay. Is Dalmatian a rock that is found in Mexico made in the shape of egg. So why I'm telling you this? Well, me amici, io sono dalmata. I am Dalmatian. For Dalmatian men and Dalmatian dogs is the same name, Dalmatinas. So that's me. Where's Dalmatia? Dalmatian is Europe. It's part of what used to be
me está recalcando la cosa, la emisión. O sea, en el parlante sale doble.
Someone talking to me? Okay. And this yellow here is Croatia. I know you Argentinian know really well because you beat us in the World Cup 3-0 in the last World Cup, but let me remind you, we kicked you out of the initial group in previous World Cup at 3-0. Okay, just to make the score even, okay? So this part is in coast is Dalmatia. I was born in a town called Split. It does not mean split like it means in English. What it means is it originates from Aspalatos. It used to be Greek town underneath there. Split is actually a Roman town built by Diocletian, who was an emperor, lived and ruled there. Let me read this person. And this is what it looks like, the town today. The actual face here is a Diocletian palace. You can walk through there. There's some built more than that. You can windsurf in one day and go ski in another day. I like windsurfing, skiing is not my trait. Okay. I studied at University of Zagreb, where I got my MD in Carta Magna School, and then I moved to, well, that's not true. I did my rotational internship in surgery, general surgery in Cordoba, Spain. Yeah. Okay. I can do that. I can do that easily, but I don't know what is going on. There's some, I hear voices. I'm thinking maybe I'm schizophrenic, but maybe I'm not. Okay. Okay, good. Okay, so I moved then to United States and I landed in Ames, Iowa. For those of you, this is United States. There is a sovereign country that Trump needs to learn. It's Canada. And there's another sovereign country below that called Mexico. And we have Gulf of Mexico. Okay, so I live here I came to Iowa State University as a graduate student working with Phil Hayden. And then I stayed as a postdoc there. And then I moved for a short time to Yale University to work with Reinhard Jahn as a postdoc. And then after that, I moved back to, no, I didn't move back. I went to Mayo Clinic, where I was a research fellow with Julio Fernandez, who is Chilean. And then from there, and I learned how to do paquete chileno if it's needed. And then I came to my first faculty position and Then I started doing nanofabrication when it was not that popular. It was the beginning of 90s, in 95. And I made my first nanofabricated device in Cornell National facility. And then after that, I actually came back to Mayo, and then I started my first faculty position at Iowa State University. During that time, I also got married in Montana. My wife's last name is Montana, so I throw a little romance. What else you can do than a namesake state on a top of the mountain where two glaciers took apart on the Logan Pass, we united and we are still there. See, it's magic. All right, so then after that, I went to University of California, Riverside, and I spent a few years. And then from there, Adriana really wanted to work with Clary Sondheimer and to do glioblastomas, and I had means, so we moved to University of Alabama, Birmingham, which we spent 17 years. During that time, this is kind of in 2013, 12 years ago. So you can tell that I expanded horizontally and that maybe aging did not treat me well. I did not treat myself well. To give you a comparison, on my expansion, vertical expansion is present in kids. Okay? I did horizontal, they did vertical. This is one of the last pictures they have because they flocked out. Wuga, my daughter, lives in Berlin. He lives in Richmond, Virginia. During the time in Alabama, I had two labs, one in Rijeka, where my wife is from. It's in Croatia. There was someone here who was in Rijeka recently. And then in Bilbao, if you speak Euskera, I'm expert. I'm just joking. And then on December 21, 2022, we embarked, and this is the actual screenshot from that trip. We went from JFK to Hong Kong, spent our Christmas there, and then moved to Hangzhou. Hangzhou is the most beautiful city in China. Marco Polo is not actually Italian. Italy didn't exist at that time. He was born what is Venetian Republic. And unlike what it says, when you're born in Venetian Republic, there's only place you can be is Venice. He was born actually on an island of Corchula, which is Dalmatia. He's my fellow Dalmatian. We are 20 miles apart born, okay? He says this is the most beautiful city on earth. Well, I think that after going through desert and all of that, he was so sweaty that when he came there and see water, probably liked it and forgot how does it look in Dalmatia. He's obviously blind. But interesting enough, Hangzhou and my born city are sister cities. So that's another thing that factoid that may have played a role in decision to go there, or may not. This is how the campus looked like, and this is how kids look like. First time in 2008 when I started exploring Chinese opportunities. Again, you can see differential expansion, and this is them in a visit to China. Coming here, let me tell you, it's challenging. This looks really nice and flat, but when you have a geoid, if you look down, if I look down, I'm almost looking at Hangzhou. I just need to go a little bit like that, and that's my Hangzhou there. So antipode, completely opposite from west to east and from south to north to Hangzhou, China, is actually Santa Fe. San Javier, Santa Fe, Argentina, which is very close to where I first time came to Argentina, to Cordoba, as Javier invited me to a school. All right, and this is what is kind of lab in the first year being there, and I'm going to show you what was the last year. We need to update that. If you wish to do postdoc or somehow come there, just join the team, please send me an email. We have plenty of positions available. All right. So let's now go to science. Is there any questions on this?
No.
Good, okay. All right, there you go. So let me just give you science now. When we think about synaptic transmission, we think about neurons. And this is here a thin section from cerebellar cortex, but it might as well come from many other regions of the brain. You can see here synapse, and you can see here in turquoise, the cell that painted, obviously, is not color EM, that is positioned to affect synapses. And indeed, these cells are astrocytes that they can listen to the synapse, whereby a neurotransmitter that is released into synaptic cleft and escapes from there by spillover can signal to astrocytes. And astrocytes can either react to that or initiate their own conversation by releasing various transmitters, including glutamate, that I will talk about, but other transmitters. There are several mechanisms of release of glutamate. In fact, there are 7 different mechanisms from astrocytes, but I will talk only about one, and that is exocytosis, because evolutionary is more preserved, and it also occurs in neurons and astrocytes alike, with some differences that I'm going to point out as we go along. Okay, so when we look at these transmitters and this signaling from astrocyte to neurons, we call that GLIA transmission. And therefore, GLIA transmitters are those transmitters released from glia cells. The reality is glutamate is actually not neurotransmitter because neurons cannot produce it de novo. They get it from astrocytes. So we refine the synthesis, not de novo, but just synthesis of transmitters good enough to be a transmitter from some cell. So these are transmitters that kind of in historic order are discovered to be released from astrocytes by exocytosis, glutamate, ATP, atrial nucleotide peptide. This serine is presently under scrutiny. In our hands, we cannot find release of these serine from astrocytes in supraoptic nucleus. It's solely by neurons, but we never repeated directly these experiments, and people are finding, people are not finding. So this is a little bit under question. No doubt that what Jean-Pierre did is correct. So I think it's just a difference between different regions of the brain. Brain-derived neurotrophic factor is one. The GLIA transmission turned out to be important for this concept of tripartite synapse, where presynaptic terminal releases neurotransmitters signal to postsynaptic cell. Astrocyte is there to... Yes?
Share your...
I haven't done anything. Okay.
Because they don't see your.
Okay, well, that's new. Okay, I don't know. Okay, please. Because he shared it before.
Yeah, this is so.
Hold on your thought on that. Okay, bottom line is, let me keep going because we'll never finish this way. So if we if you now look at what is glial transmission important, it turned out to be important in sleep. It turned out to be too important in learning and memory. And we did work on gut, in which we showed that one way of releasing ATP through connexin 43 was important for motility of gut. While, for instance, exocytosis, which is in different part of the gut lining, was important for secretion from the gut. Okay, so no problem. Okay, can I
Ya está compartido. Yo lo estoy viendo por acá por el Zoom, ya está.
Yeah. All right. So let's. I apologize those on the website. Well, I cannot advance. Hello, I cannot move. It's locked up. I cannot move the slides. How?
Use the arrows.
I did. Ah, okay. Okay, good. I have too fine touch. I have my feminine side when I'm touching buttons. Sorry. Okay, so this is what I told you when we are off, and here's Javier. Javier.
It's dead touch. It's dead touch.
Which
Click there.
there?
No, I click there and then I go there.
Hold on. You click here.
I click. Yeah. I put the arrow here.
Okay, all right. Okay, all right. Let me try this. Okay. Okay, so let me lead you now through at least first set of experiments that we done, and that is to this is some time ago where we use here I used here an island of astrocyte from visual cortex in which I added bradykinin. Bradykinin is actually endogenous signaling molecule that is generated interface between the brain parenchyma and vasculature. And it acts through bradykinin 2 receptors, and you can see there is an increase of calcium, which with the time resides to the resting level. In parallel, if you take and collect supernatants, you can add bradykinin, you can see there is an increased release of glutamate. With time, we show that calcium increase sufficient necessary to cause glutamate release, which is a footprint of exocytosis. And the mechanism is definitely exocytotic because you could cleave with tetanus toxins and synaptobremin 2 and negatively modulate with rose-bengal V-glutes. Let me show you these molecules that I'm talking about. This is the present picture of how exocytotic machinery looks in astrocytes looks like in a abbreviated form, and it's done mainly by these 4 laboratories. So you see here a vesicle that has glutamate, and it's anchored to the plasma membrane, snared to the plasma membrane by proteins called collectively snare, which is a really nice acronym that describes what they do. And astrocytes having synaptobremin 2 and 3, hence very similar to neurons, and have a ternary complex formed with syntaxin 1A, same as in neurons, but SNAP23 rather than SNAP25B, which is in neurons. And here, synaptotagmin 4 has a function. We really don't have published data yet on what is that calcium sensor. We know it's one of the tagnins that is found in neurons, but we haven't published it yet. Synaptotagmin 4 is not calcium sensor in mammalian system, and is in drosophila. Okay. The other set of proteins is here, and one is VATPase. That expense of energy of ATP pumps protons inside the vesicle, makes this croton gradient that is 5.5 pH inside as opposed to 7.2 in cytosol. Well, it's a small number in a small difference in number, but remember that's logarithmic scale. It's huge, okay? So then that proton gradient is used by vesicular glutamate transporters 1, 2, and 3, all expressed in astrocytes with associated chloride flux to bring glutamate inside the vesicle at expense of slightly deprotonating a vesicle, which then ATPase takes care of. Okay, so let me tell you how did we actually do those experiments. We use so-called solitary astrocytes. We do that from visual cortex, be that from Sprague-Duley rats or CB57 mice or human astrocytes that we cannot choose where it comes from. It comes where it comes from, okay? And then, I mean, region of the brain. And it comes actually from patients that have traumatic brain injury, and you have to do a little extra resection. If there's any neurosurgeon will tell you that in a room. And then we take a little piece at the periphery, and that's where we get our human astrocytes from, or we use it for glioblastoma cells. Those are those malignant astrocytes. Once we look at astrocyte like that, obviously convenient here, this is dead astrocyte here, stained for GFAP and country stained for DAPI. Normally we see them, it's a purified culture. Basically everything is astrocytes. You find a cell of interest. You bring a patch pipette in the field of view to induce mechanical contact that electronically you look at, there is an increase of resistance, so you know how much of pressure you apply. And there's a calcium increase, which we can detect, I showed you earlier, with a calcium indicator. And then we measure glutamate release. Glutamate release, we can use glue sniffers. Of course, we are not stupid, but the issue is that if you start an exocytosis and you deliver in a sensor with exocytosis, you might not back it off and do some other way first, how you can measure glutamate. So we do it with glutamate dehydrogenase, which we purified from bovine, and we put it in add some NAD+. In presence of NAD+ and glutamate, GDH converts glutamate to alpha-ketoglutarate with concomitant conversion of NAD+ to NADH, which is a fluorescent product when excited with UV light. So based on accumulation of this fluorescent product, we record glutamate release. Let me show you how does this look like. This is a solitary astrocyte loaded with calcium indicator. When one touches here, you can see increase of calcium, and with the time, it subsides the resting level. Now you're looking at NADH, NADPH, because there is some autofluorescence from cells, from mitochondria. They have them. But we don't analyze that signal. What we analyze is this halo that is around cell. And you can see how there's a appreciable increase of fluorescence in surround as the glutamate diffuses through the extracellular space. This is the summary of this mechanically induced calcium increases and mechanically induced glutamate release. It is calcium dependent because if you use BAFTA-AM, there is reduction of calcium level with reduction of glutamate release. Given there is a calcium dependency, one always suspects there might be exocytotic/vesicular release. In order to assess that, we use 3 different compounds to tamper with vesicular proteins. One is we can cleave it with holoprotein of tetanus toxin. Because any of this is downstream of calcium, so-called, we are not affecting calcium levels, but we are potentially affecting glutamate release. Either that treatment or additional 2 treatments where we use buffelomycin to collapse protogradient and not allow glutamate to fill the vesicle, or by rose bengal to block basically vesicular transporter, we have reduction in glutamate release. In fact, with rose bengal and 0.5 micromolar, and 1 micromolar should be full dosage we should use. With half of that, 97% of that disappears. Okay, in order to mechanical stimulation, you may ask yourself, Vlad, you don't shish kebab the brain. Okay, well, that's not attempt. Okay, the intent is to gently touch and stretch the cell to emulate what astrocytes might be experiencing in an interface with blood vessels, where every pulse that it comes in, they stretch. Okay, but anywhere to assess the different stimuli, we use stimulus with bradykinin and ATP. You know this fellow. And ATP is acting, this is GPCR. This is GPCR and ionotropic receptors. There is an increase of calcium. It's unaffected if we treat with very low dose of rose bengal, and we have it in release. So 3 different stimuli, ATP, bradykinin, and mechanical stimulation, all tell you the same thing, that this is exocytotic vesicular release. Well, then people tell me, well, if you look in Palais Atlas, you cannot find vesicles. Actually, you can if you look carefully. I can give you pages where you should go and look at, but I don't need to go to Palais because I need to read a little bit in French or Spanish or Italian and find out that in 1910, actually Najot, the physician urologist from Paris, has described in Royal Society address these granules that he's seen in astrocytes. I mean, you can read this. It's a great substance, GLIA, which astrocytes from rabbit. So here you can see these that I think he described for secretion that we know. And then he goes in a strain last 2 paragraphs telling me how this is fractions of mitochondria being expelled from the cell. But even that happens as a cytotic. So something more contemporary I need to offer you, okay? Rather than this is confirmation of what I found, or I confirm him, whatever. So here's another thing. If you put astrocytes in so-called recycling dyes, FM143, FM46, FM210, RH404, you name it, I care less, we try them all, and you stimulate with bradykinin and you wash it away, you should have some puncta, and we do. Okay, then if you look at an EM, and this is a border with two astrocytes, you should see some clear vesicles, and we do. We can see here a merger with the plasma membrane. You can even see omega shapes here, consistent with pinching off and fusion of vesicles to and from the plasma membrane. Okay, well, if this is all, you would laugh at me. Okay, I need to show you something better than that. And what I need to show you that we can actually real-time image vesicle fusion in astrocytes or vesicle fusion. So we do that by using so-called superecliptic synaptofluorine. It's a big name for basically something that has a really nice quenching capability. It's a chimeric protein produced by, first time by Gero Miesenbock, while he was in Jim Rothman lab. And what happens is here, he added EGFP, which is by itself pH sensitive. That's from Aquara victoria, from a jellyfish. And then he genetically modified it to have a better dynamic range. So 5.5 is nearly completely quenched or eclipsed, therefore superecliptic. And at rest, that's about 5.5 pH. And then when a vesicle fuses to the plasma membrane, what you have is you have deprotonation, and this lights up like a Christmas tree, and then goes and endocytosis and reacidification, and then you don't see. So let me show you a video in a moment acquired from these cells. This is a normal microscopy that has a 60x objective, and you can look at here, individual astrocytes in culture. What we're gonna do, we're gonna juice it up. We're gonna look at only part of that, because what you're using is total internal reflection fluorescent microscope, also called a Van Essert vein microscope. Let me briefly explain how does that work. I guess I skipped that slide here, sorry. And that works in this principle of reflection, in which if you provide light to the interface between high refractive and low refractive index and exceeds so-called critical angle, the light is totally reflecting, at least it looks so, and that's why it's called total internal reflection. But one consequence is that some light does go through and forms a Van Essert wave, which exponentially decays of that interface and penetrates within 70 nanometers. So we are looking thin sections of astrocyte facing the glass and equivalent to the ultra thin section of EM. Okay, so let's look at that movie. If there are any vesicular fusion, you should see them here like little popping, like popcorns under bradykinin stimulation. Okay. So here we go. Okay, as you can dance. All right, so my wife said I shouldn't do that because I look like an elephant in glass manager. Okay, glass shop, okay. If I use some weird language, just let me know. Maybe I incorporated from Chinese. But Wendy will be here, lector for my English. So here's the thing. What we're going to look at now, let's look at the time course and spatial distribution of that. I told you that SNAP23 is expressed not 25. So here's the first surprise. In neurons and in chromaffin cells, if you have SNAP25, you have so-called burst of release, which is shown here. But it's very quick. It's within microseconds. So we have here a lazy burst. It's a 3 to 7 minutes. In mechanical stimulation, we have a burst. What governs that? We have no clue. But it's not SNAP25B because we try to find it with anything under the earth, including, protein sequencing, anything else. We don't have a single copy that we can find. All right, so maybe there is somewhere there, but it's not for us. But we see this envelope with bradykinin, with ATP, with ionophore, which rise only calcium inside the cell, or spontaneous, or so-called laterotoxin that squirts out those vesicles, laterophilin, sucrose that squirts them based on the osmotic pressure. But if SNAP25 causes in neurons that burst, we thought maybe we should just add SNAP25B overexpressed up to wazoo and see that it converts this to a burst, but it doesn't. So it really is something different. is involved here, which we don't know what yet. So you can keep in mind there is a burst and there is a sustained release, like what we call in chromophore cells an envelope. But for those of you with the trained eye, you can see actually you can see 3 different events in that movie. But I'm going to focus on 2 that are major ones, so-called kiss and run. And that's a Jacopo Maldolesi named it. So don't blame me, okay? Jacopo sees this as a kiss and run and full fusion. I like the terms because then I can insinuate things. Okay, I can say that this kiss and run is like a little peck in the cheek. Okay, full fusion, that's like a French kiss. The vesicle comes down, completely collapsing into plasma membrane. And you can see how it's different type of message that you might be delivering, right? So that's what astrocytes use to their advantage, delivering a different message there. But I think it's very important when you go into French kiss is retrieval or in kiss and run. And you can see because maybe it's a kiss and stay, peck in the cheek 1 second is one thing. But if you stay longer, you might be sending different message. We'll come back. Let me tell you how we do this. We look for spots that statistically are 3 standard deviations above the background. We claim they are full fusion if there is an increase of fluorescence with appreciative increase of spread of that fluorescence or width of the signal. Transient fusion has no increase in full width half max, meaning it stays locally there and pinches off. When we do so, we see 2 types of fusion. One is that we see those 2 types of fusions, and it's conveniently 50-50 spontaneous. I did not expect that, but there we are. And using different stimuli, we can swing it to the left, which is shown in blue, and to the right, which is shown in red, to follow our beautiful country parties that we have. That's just, I have to do that, okay? And bradykinin is causing transient fusion. Bradykinin is causing mostly full fusions, while touch is mainly transient fusions. Okay, so the message is that different stimuli govern which type of release we have. Now, in the kiss and run was like they look long. So we studied how long is this kiss and run. They're mighty long, 1 second to 2 seconds. Which with the fusion pore that we know what exists when they fuse, the entire content of glutamate is gone. It diffuses the same way as it would diffuse on the same time scale from these full fusions. And I give you here tau so you can look that it's within the same domain, which means that it's really into maybe not just which message you send, but what is that, how quickly you can retrieve from there. So that much about timing. Let me show you a little bit about space, right? The issue is, where do they fuse? I mean, in neurons, we say there are fusion sites. Well, let's see where astrocytes have fusion sites. The first order of business is to demystify or see what is the size of vesicles. You see papers occurring in very high-profile journals saying that a group of astrocytes, which was 3, a group of vesicles, 3 vesicles, emphasis, are there, and they are 30 nanometers. Well, 3 vesicles for me is anecdotal, okay? So we took 23 cells and looked at 8,000 vesicles, and we used the sectioning power of TURF. What that means is that if we put beads of known size, they're calibrated for EM and their fluorescence, we can build an expected sigmoid curve if we illuminate this first with TURF, then we change the angle to what would be a normal microscopy, and then we make a ratio. And physical chemistry teaches you that's an exponential curve. Then when we do the same, and these are like beads. You can see here bead. If it's here, it's less illuminated with TURF, and it's more illuminated if you have normal trans illumination. Okay, so now we do the same thing if we take not fluorine, but we put GFAP to have a little bit more fluorescence, and we look at vesicles, and we put them then on this curve, and voila, here we go. There are 161 to 422 nanometers, which these are large vesicles, 300 nanometer, but on EM they look clear. So this is not as in neurons, it's a little bit larger than in neurons, but 10 times in diameter, which in volumes you can imagine. But then, you know, for some people, this is a little bit odd. You know, that we need to do this ratiometric approach. So we did it a different way with Robert Zorits. We used structural elimination microscopy, which straight up has a resolution to bring us down to 100 nanometers, and using less astrocytes, but certainly more than 3 vesicles, then we have 305. So very close data. So therefore, these are vesicles of 300 nanometers in diameter. The next thing is to look, where do they fuse? And we can do that by using density mapping, so-called. Here is the movie. No music now. Okay, so, but it shows you there the kiss and runs fusing in blue and in red full fusions. And you can see what appears to be preferential fusion sites. If there are multiple fusions, you have rings around. And statistically, yes, we do have inhomogeneous density here. So therefore, there are fusion areas. Now let's look at what the fusion areas might be. The way to do that is to make so-called near neighbor analysis and also replicate. Let me explain it. If you go near neighbor analysis, what we find out that the vesicles like to fuse where they previously fused, shown in a red bar, and that's most incidents there. But the next mode is variable, but it is kind of dependent of the stimulus, and it can vary from mechanical being all 3.5 micrometers apart to the smallest one being actually with the spontaneous at 1.64. We measured this by 6 next near neighbors because power analysis told us that that's what we need to do. And now let's look at a replicate. A replicate is a model that is established in forestry to look at, is there any differential density of trees? What we did is we obviously, we have to adjust it for Flavia for Brazil, because you have canopies in Amazon. So we don't have one tree, you have trees growing on top of trees. So we adjust it that way. And it is the same method that the New York City Police uses to find where's the crime in certain skyscrapers. So what we've done now is we are limited by the radius of the image. And if you look at throughout the radius, you can count how many vesicles are within there in real sample, make Poisson distribution of that simulated, showing here 99% confidence interval in gray, and ask in our real sample, are we below that, which would be anti-clustering, or are we above that, which we are not here, but here we are? That would mean that we have a clustering of vesicles. So we have clustering vesicles here with different radius for different stimuli, overall about 3 micrometer radius, which brings us to looking at this mess in astrocytes. This is actually a creative mess. If you do statistics, we find that there are actually functional fusion zones. There are 6 micrometer diameters. There are a certain number of vesicles within 300 micrometers, and there are 2 micrometer pulp. Okay, now that's much how I can show you, and this is something that we await to still to publish. Now, calcium. Calcium is important. There is a Hill coefficient of 2.1 to 2.7 that we measured, which is similar to neurons 2 to 4, and which means that we have Tagmin as a sensor with still it's unpublished. Okay, that calcium comes from multiple sources. Let me summarize here about a dozen of papers in one slide, telling you that most of the calcium comes from intracellular stores from smooth endoplasmic reticulum. From there is recruited by IP3 receptors and RNA receptors. The store is filled by circuit pumps that draw in calcium, of course, from the extracellular space. In our hands, that involves 2 entities, transient receptor potential C1, canonical 1, and also reverse mode of sodium calcium exchanger. As this calcium rises up or going down, the dynamics are buffered by mitochondria, and that is done by MCU, mitochondrial uniporter, that takes calcium in, and it's released with 2 ways. One is with mitochondrial permeability transition port that, yes, can by flickering release physiologically calcium, not release big molecules, and NCLX, which is a different entity than NCX, in exchange of sodium. This calcium then triggers snare through Tagmin snares to release glutamine. So that is what I gave you I guess somewhere I lost it. I didn't present this slide in all that commotion, okay? What I show you in background, tripartite synapse exocytosis and evidence of a cyclic exocytotic glutamate release from astrocytes and gave you calcium overview. So I want to shift gears now to pathology. What I want to show you is the evidence for exocytotic glutamate release from glioblastoma multiforme. Let me be very clinical. When I say glioblastoma multiforme, I only look at one thing, and that is by astrocytoma 4 by World Health Organization, by European Association Neuro-Oncology. It has to be IDH wild type, not mutated. The one with the wild type is the best killer there is. It kills people in 14 months. The one which is mutated, it gives people to live longer. So we are working on the glioblastoma multiforme, which now that pancreatic cancer is essentially cured, but needs to wait time to increase statistics. We are going after the one which is the worst one that can be, one can have. Okay, the bradykinin plays a role in an invasion of these cells that were shown by Vedrana Montana, working with Harry Sondheim. What she's done is rather than using inserts, they have different matrix that we've seen before yesterday. She used a little bit better fabric. She used acute slides, so you have a real fabric of the brain. Put the green human glioblastoma cells on top of that and ask how they penetrate parenchyma when they do that directly, and how they do that if they go along the blood vessels that she can stain in live slices. Bottom line is that in presence of bradykinin, they penetrate deeper into tissue. It can be blocked by bradykinin 2 receptor blocker called HOI-140 or icatibant, same different. And then I'm going to tell you a third name called firazir in a moment. But this is the same peptide that blocks that. And then she looked also how much does it associate with blood vessels. So if you have bradykinin present, they're associated in more blood vessels, and that's how they crawl into spaces to invade. If she uses icatibant, it's interesting finding. She not only blocks that, but it goes below that. The simplest explanation to that is that in these slices, there's still production of bradykinin and an interface between blood capillary endothelial cells and brain parenchyma, in this case with astrocytes or glioblastoma. And same result she got if she used a short hairpin for bradykinin receptor 2. Okay, so now that we know that, what the hell glutamate has to do with that? Well, it has to do that, again, Harry Sondheim has shown that AMPA receptors are important for invasion of these cells into parenchyma. And he found that SXC has a role of that in vitro, but failed in a clinical trial. And I show you later why I think a clinical trial failed. To me, intuitively, this cannot be only thing to play a role. Because when I look at the patient charts, there are almost half of patients that are knockouts in their glioblastoma for SXC. So the protein that doesn't exist could not possibly govern the release. So I thought because evolutionary, again, exocytosis is more is earlier than SXC, I thought that it might be the way that it does work on both. So what we've done is we use now what is called patient-derived xenolines. What that is, you take it out of brain and you propagate it. And this particular one, GBM14P, has been made by James Sarkari at the Mayo Clinic 17 years ago. And every 10 propagation, we look at that they have the same properties in patient. That one is a knockout of the SXC. The other one is this GBM22P, which is actually expressing gloriously SXC well above the normal expression. Regardless of that, if you put bradykinin, they have similar glutamate release, and we can similarly inhibit it by bradykinin. Okay, the way that you propagate these PDXs will become important in a second. You never have any serum present. Once you have serum, you can discard them. They're growing without serum in the dish. And they're growing in the flank of nude mouse or humanized mouse, so they don't get rejection. Otherwise, there's immune system responding. And so one caveat of this that we don't have normal immune response that normally people have. So that we cannot test. That's the only possible really in human. Right? We can take it out from here, put it in a dish, put it back there, or put it inside endocranially, which is entopic versus ectopic expression. Okay, let's go forward now. If this is true, what I told you, then we need some confirmation for Rose-Bengal block that. Well, we then use sRNA, cleave synaptobremin 2 or down expression, and you give less release of glutamate. Okay, so that works. And you can say, well, Vlad, you have a molecule here that is fantastic. You can do translational research. I agree with you, but there's nothing FDA approved for this. And I'll show you what we're doing in that respect. What really caught my eye is that Firazyr is a preparation for human icatibant that is approved by FDA and Chinese FDA, US and Chinese, for use as a bradykinin 2 antagonist in something called hereditary angioedema, where people have this all skin looks like someone put a 1-inch steak and they need acute treatment. So this is treatment that you receive one dosage. We propose to use this in glioblastoma as off-label. Okay? And you can see that bradykinin in presence of OA140 in this case is not really Firazyr because we don't use Firazyr here. This cost $10 and Firazyr cost $10,000. Okay, one shot. Okay, it's the same thing except this cannot be given to human. So you can see there is a nearly completely obliterated glutamate release. Okay, now the problem is that a company that made that Firazyr claims that Firazyr does not get into brain because of blood-brain barrier, and only occasionally patients have headaches, which means it might pass. But the resolution is very simple. When glioblastoma is there, you don't have normal blood-brain barrier. You have neovascularization, leaky blood-brain barrier interface. So therefore, maybe he's getting there exactly where we need it, but we need to somehow visualize. In order to do so, we synthesize icatibant with tamra, which is kind of a rhodamine spectrum, which is a fluorescent product. And we applied now this to, so we had it, and we want to know now, does it, addition of that molecule affect the peptide? The short answer is no, and here's the proof of that. Then when we load with human glioblastoma cells, we raise calcium with bradykinin, and if you put tapra icatibant, we completely block it. So it works fine. It also labels cells. The cells that I show you now, D45 and PDX is 14 and 22, are all higher expressors of bradykinin receptor 2 that is normal human astrocyte. There's a criticism from syngenic community. What means is people who work on rat glioblastoma in rat and mouse in mouse. I have to tell you, those are not normal glioblastomas. They're generated by irradiation cells and made and then put back in rat brain as a model. They're not good model for human. Let me tell you why. Mouse work tells you that bradykinin receptor will cure human. Well, it won't because there is no significant expression of bradykinin receptor 1, but there is a bradykinin receptor 2. I'm going to come back to that in another slide. So I just want to let you know, mouse glioblastoma and rat glioblastoma created in 1945 during just after World War II, they are not good model for human glioblastoma. Mouse and rats are not small human. So what we do now, this is now a mouse brain, obviously, and drawing. And this is a section where we injected the cells and they formed a tumor. And now we can investigate where did tamra icatibant go. Well, it did not go to extra and on contralateral side because there is no label. There's no tumor there. There's no label. Argument is maybe could go through area postrema, but we didn't check that. These are blood vessels. But on the side of the tumor, you can see there is here mostly tamra icatibant locates together with the body, but there's some extravasation, although it's not. I mean, some illusion to the parenchyma. With both places have extravasation because some is within, some is outside of vesicle. So that's positive for us. That means that tamra icatibant is getting delivered into the GBM tumor mass and some peritumoral tissue exactly where we need. Okay. Does it affect now the real glioblastoma? We put now human glioblastoma into nude mice for a good measure. We don't use only green ones, we use red ones. We let them develop to the tumor, and then we start treating with retroorbital injections that are equivalent to human subcutaneous dose, so allometrically adjusted. We wait until they have morbidity criteria for sucking, or we wait until, well, we don't wait. Occasionally they die by themselves. We find them dead. We use equal number of females and males. Reason being is that glioblastoma is incredibly discriminatory to male. It kills males much better than females. And the therapies that we have treat much better females than males. So it was the idea here that I thought that same pattern would be followed by icatibant, but it didn't. Icatibant did not care. It has same effect on female and male mice. And the bottom line is that we extended the life of those animals by 35%, which, let me put you in perspective. It might not be much, but in the last 25 years, we have 10% increase in life expectancy on all treatments that we thus far generated. So this is pretty good promise. Okay, we looked at the volume of slices, and these tumors do not demarcate well on magnetic resonance imaging, MRI. In mice, we can see them clearly in MRI when they all bulge out of skin if the mouse even survives. Okay, so the way to do it is old-fashioned way, poor man's way too, but the way it works is you slice the slices that you know exactly 300 micrometers, you measure the volume, you generate the volume area, you generate the volume out of that, and you find out that we have 58% reduction of volume. So what I show you thus far is that it looks like we can pharmacologically tame tumor progression in vivo. And can we do that in human? So reality is this is the first thing I've done, and I told you a story a different way. First time I've done this, so I don't repeat the error of spending millions of dollars and going into crapshoot in 10 years of work, okay? The reason why I'm saying this is if someone did this, would not go into trial for SXE. Okay, so let's look at what we've done here. We take gene expression data. This is TCGA library by NIH that has RNA-seq data, and we take human patients with GBM, bona fide GBM, IDH wild type, and we look at if there are high expressors of that RNA, top quartile, versus the rest of 75%, is there any difference? Well, if you have high expression, low expression of SXE, there is no difference. It's essentially the same. So if you treat patients, why would you see the difference? Let me give you another one. 4 clinical trials failed with the MMP2 and MMP9. Well, no kidding. If you do this, you will spend $8 million. So here we go. It doesn't work in human. It may work in the mouse, but it doesn't work in human. Here's a mouse example. Mouse syngenic model tells me that bradykinin 1 is important. It treats animal. The animal is as good as it was. Well, it doesn't matter in human because there is no difference between bradykinin 1 and 2. I understand this is mRNA, but I'll show you in a moment some protein data that we back this up. Out of proteins that evolve in our potential pathway, not everything is useful. Look at this, VAMP3. Doesn't matter. It lights up like a Christmas tree. But you look at the cousin of that, VAMP2, very similar protein. In fact, in neurons, you have synaptic transmission. You knock out VAMP2, puts VAMP3, and you have synaptic transmission. But in these cells, it makes difference. So it's interactive molecules that we are looking now with the BioID, which is inside the thing from here. But look, there is a significance in VAMP2 is the highest. And then bradykinin 2. Let me now show you how does in a human look at bradykinin receptor 2. This is the data, again, RNA data. Survival time is much longer if you have lower expression of bradykinin receptor 2. What that is these people here as opposed to these people. So we cannot treat everybody. What we can do personalized medicine, such as, let's say, in breast cancer, is it less important to know whether a woman has a hair or man, because you can have breast cancers in men, HER2 or plus minus. It's very important for clinical findings. So here's the bradykinin receptor 2. We can treat part of the population. By RNA, 1/4. By protein, it looks like it's maybe half a population, okay? This is the tissue from humans. This is a normal brain. This is the GBM. Some patients will just have the same level. You cannot do that. And you can treat these ones, okay? VAM2, that was really shocking that this is the highest, the best target there is. There's a little complication. If you go back to 2004, we show that neurons do express about 100 to 1,000 times more VAM2 than astrocytes. Well, what a surprise then is to find that astrocytes here have about 100 times less mRNA. That's expected. What was unexpected is that these high expressors actually live shorter than the low expressors. A student in the lab now is making Vaccinia viruses that we know are GBM tropic, and those are oncolytic, but in the first few cycles, we actually block release of glutamate, so we have less glutamate when we lyse the cell. It's look very promising. In animals, we'll see that will be really slow ride to get it in human, as you know, but we do have company that can push that to, once we have all our stuff together, maybe in 3 to 5 years. Okay, so this is actually pretty interesting because I've done this myself, being bored during the COVID. I looked myself this with RNA-seq in so-called IV glioblastoma atlas. And I looked at, we defined, this thing is on our way here, but actually not everything that you need is important. Arbitrary in clinic, we divide tumor in 7 different parts. And I'm showing you here something that you've seen already, that bradykinin receptor 2 is more where hyperplastic blood vessels are. You saw that in that confocal image, and microvascular proliferation. What is interesting here that VAM2 is there in vicinity at the leading edge, and infiltrating tumor. So what that tells you that maybe you can combine these 2 adjuvant therapies. They're all adjuvant to what we have standard of care. And have, additive effect, perhaps. We'll see. This is what I showed you, that we are addressing the role of bradykinin 2 in GBM cell invasion, that we are now poised to define the extent of differences between human, person to person, and within the tumor for diversity of these cancers. We are now in a phase 0 and 1 clinical trial with the patients that undergo so-called magnetic resonance spectroscopy, where you're looking at the signal of glutamine and glutamate in these patients, and we inject icatibant. The ones that have reduction of that signal are potential patients when we remove the glioblastoma. Then it's checked again for content, if that matches, and then might actually receive it later for a treatment. As I told you, it's mighty expensive treatment. $9,800 cost me in pharmacy at UAB when I left, which with present inflation after these wonderful economic developments in the country, probably cost $12,000. But what it is in China, it costs from the same company in Germany, imported to China, same preparation costs $750, which brings me to ability to actually do clinical trial on larger scale with the same amount of money. Still, it is, for those of you who are physicians, injecting peptide is not fun. We know that from insulin, which actually works really well, but bottom line is we are looking for small organic molecules that bind and have similar Ki. In fact, these ones that we synthesize now, 3 of them have an order of magnitude to 2 order of magnitude better Ki than has icatibant. And calculating now, it would cost about $10, which is not bad. So similar to what I told you earlier, icatibant that we put, that is dirty icatibant for mice. The other thing what we're doing is structural relationship to bradykinin receptor 2. We know what is the X-ray crystallography, and we can go now in silicon using computational biology, look at the compounds that are in China digitized and deposited into libraries that belong to so-called traditional Chinese medicine. And those are, I'm looking at no voodoo dolls, but I'm looking at the plant and animal compounds that might act upon that and might end up being cheaper, or, we already know at least human exposure to it, as opposed to something that we synthesize in the dish. We have too many hits, that's the problem, and we're trying to prune down that. I thought there would be none. Unfortunately, there's plenty. And plenty is always a problem because you need to reduce what you're gonna test. And finally, I show you that there is no difference between sexes, which is one more promise of this. The final note is there is a library in China, like TCGA in States, that is CGTA has much more patients and it's Asian population. This bradykinin receptor 2 is the same working in Asian population, which is not the case for all targets. So we might have something that will work, we know for sure, in Caucasian population, Afro-American population, and Asian population. And that's where I'll stop. This is obviously people are from different era here. Vedrana Montana is actually in Hangzhou. Xue Hua, Yin Chun-Li, Eric Malarkey, Rino Reyes, Manoj Gotipati, Chantel Hopkins, Alexis Smiley helped in this, Andrei Naegin NSF, and Robert Zoritz and Verhadsky, Alex Verhadsky collaborated. Ming Gao and Jake Chen are involved in computational biology. Chris Wiley is radiation oncologist. Anita Gemelan works on so-called brain tumor initiating cells or stem cells that we are really keen on. I didn't go into that. And that continues now with this is all picture of the lab again in September. It's different composition. Yeah, it's different composition. We still have 2 males, but different 2 males, but the rest is all female lab. That's because Chinese women are more curious than Chinese men. It's true. And it's funded under Chung Kong program and Kumpen Action. And if you would like to join them, please send me an email. And thank you very much. And sorry for all that commotion initially. Thank you.
So we have thank you very much. We have time for 2 or 3 questions. Okay.
I think you need to come somewhere here.
Or
Maybe try not to stretch it too much, because whatever you do here is finicky. And when you go, don't step on cables when you're here.
Yeah.
Okay. Thank you very much for this presentation. And I love exercise. There's this paper from Bradley Zuckerov, published in 2022. And he used this iBot animal fused to CNP3, a CNP3 animal with the iBot to prove that the exocytosis of the oligodendrocytes, that the oligodendrocytes promote the exocytosis of binding components that found the myelin membrane. So when I was hearing your talk, I was wondering if you try or if it makes sense to try the iBot animal with a GFAP3 to remove the BAMP2 and 3 and inhibit the exocytosis in your conditions.
Yeah, it makes sense. So something that we are planning. You can hear me, right? Yeah. Something that we are planning to do is actually to even look at more than that, is what would be oligodendrocytic contribution in an environment around the tumor. We have a little problem with IBA1 because we are using now the immune compromised mice, right? So we can try that. But you know, when an immunocompromised, this is a leaky blood-brain barrier there. And you would suspect that, as anything else, that killer cells and everything else would invade that. We're trying to resolve that, okay? Another thing is that I think mouse is not good enough. So we are now trying to use primates, and we have we don't have it now because Anna Wang decided to move from China to freaking New York, okay? But we will have some of those that are equivalent to nude mouse, that are nude monkeys, primates, and we'll use some of that, but it seemed not to be quite necessary if you're careful with that. Looking at library and not going like, based just on animal and not look at the human data. There's plenty of human data now that you can prevent failures. But I think that will be one direction that someone in the lab taken. And actually yesterday was eye-opening to me how much stuff we should be able to do with oligos. Because I was not too certain, yesterday actually, you guys sold it to me, some little tricks there of the trade that I think would be quite interesting to look at, especially if the tumors go into like internal capsule and some of those pathways when you have so many oligos. Like when they go there, and those are easy to test because if you go extrapyramidal pathway, you can test it so simply in clinics. With Babinski sign, if that means anything to you, that one is well known and others. Patricia, just yell. Everyone can hear you. Look. Yes.
I was amazed for the spatial organization that you show us about the kids and run
Mm-hmm.
and the other.
Mm-hmm.
I don't remember which kind of model was, but it has related to the relationship of the sub-regional of the exercise to the blood vessels of the synapse, or has some relation with
That has no relationship except with other cell. Okay.
Then we are isolated.
So those are isolated astrocytes grown in carpet or grown to monolayer, and there's no any relationship to neuron. Now you're going to tell me how simple it would be to put neuron. It's very simple. It's very simple. It's piece of cake. The only problem is the end of the astrocytes are thin, and people say they are so thin. Well, yes, they are thin unless you come to nano world. When you come to my world of 70 nanometer, I can see that membrane, a second membrane up there where the synapse is. So I'm screwed. So we are looking to do that a little bit different. If we manage to do, and they're still lacking probes, it's getting better. But and you probably know I had paper with Stefan Hell, so I have a first-hand, insight to STED. And it might be that we can do it with STED within next year or so. We'll see. We'll see.
Second one.
Yes.
This is something related to what I'm doing. It's been shown that when you metabolic use the glioma with fluoroacetate, gets a better performance, a paper in science that I don't know whether you have any evidence that the mitochondrial metabolism has something to do with the exocytosis and after
Please, you must be. Do you have a relationship with KGB? KGB? You have some relationship like KGB or so. Those cameras in my lab or something, aren't you? Yes, the answer is yes. There is a big change in OXPHOX and there is big change in glycolysis. In glycolysis, nothing unexpected. They go over time in Warburg effect. No matter which ones they use, it's interesting when you challenge them with lactate and glucose, and we do that with normal lacticemia, normal glucosemia, which is arbitrary 5 millimolar of glucose and 2.5 millimolar lactate, which we can argue whether it's normal. Okay, it's normal if you move. If you lay down, it's 1 millimolar. So then we increase it lactate as we would have with the exercise. We decrease lactate as you have the depletion. We increase as you would have diabetes, not coma, but close to it. And we decrease it as you would have it when you have dumping syndrome. And and it's affected, absolutely. And it's very interesting. It's affected a few proteins only. So we are now going toward that direction. We're going something I hate. And we're doing seahorse, which I think is complete crap of shit, but everyone in the lab tells me different when good for you. And as my late friend Arnes Kochbo would say, it's complicated, and I only said it's crap, okay? And the reason why I'm saying this because it does give some insight, if it's confirmation of something else you have, but that's going to lead you for discovery. I'm sorry to say, I have never seen. It's not flexible enough, and I think it's just when you look at what does it measure, it measures that. You know, it measures the proton flux, or measures, you know, OCR, you know, extracellular oxygen, but it's not directly measure glycolysis. There are better ways to do that. You have you have now glucose sensors, you have pyruvate sensors, you have LILAC, which is lactate sensor based on film from Gary Yellen. You know, you have a bunch of these that we intend to use. We haven't used them yet, okay? And we intend to use them. We have them ready for in vivo expression with ITR2, so it's AAV2, and probably we're gonna put caps at 5 or 9, whatever. We're gonna think about which one is better. Probably 5. But yes, we are planning to do that, and but proteomics and transcriptomics give you different hits, but when you take common hits, it crystallizes story that with lactate. And it's very much along the lines what we published in neurochemistry research some time ago in 2022. The lactate is shuttling, lactate somehow is shuttling in at the level of citrate that the cycle does not continue to alpha ketoglutarate and glutamate, but rather it goes to fatty acid synthesis. And that is common between astrocytes and glioblastoma. The other stuff that we published that affects intermediate filaments is not a case in glioblastoma, which is very surprising to me, but because I don't know how they crawl, but so that's something that we should look at now. That's that long winding answer to your question. And that comes, you know, I'm logoroic when I go out of China because finally I can speak English.
And last one.
Yeah.
Thank you for the talk. Maybe the simple question, but I cannot stop thinking about it. How does morphology switches the form of the vesicles? I mean, if you have reactive astrocyte or a swelled astrocyte, does have to be reactive.
So let me make a disclaimer. Everything you see here is reactive. We took it out of the brain, we mash out the brain, go through pipette, put it down, and it expresses glorious GFAP. That is reactive astrocyte by any definition. So what we see is that vesicles are, if you use dibutyric cyclic AMP, yeah, they're distributed. And they're always on processes, but there are some that they cycle more so in body. And it's kind of pinching off directly of Golgi and goes to plasma membrane. Interesting in gloves, that is dependent on, which I didn't go into here. We have a whole study on connexin 43. You go back in 1994, Mike Nedergaard published a paper that there is astrocyte neurosignaling based on connexin 43. While everyone has forgotten about that, and everyone was trying to create a fight between two of us, we are friendly, and we understand the difference in preparations. So I took upon looking at that, and it looks like if you knock out connexin 43, then the preferential fusions are only there on endosomal pinching off. If you have connexin 43, it goes throughout the entire cell. And you can see, and that is how maybe connexin 43, aside from one set of experiments, this we need to understand the difference, how it might contribute. So that one indirectly tells you it's not only shape, but other molecules that can shape where these. So those astrocytes have exact same shape. We cannot, wow. That's the best statement. I'll take that back. They have exact same shape if you use morphometric analysis that we created that is more sensitive to the one you can buy. And that is based on tracing the thing, but tracing the thing, this is like my kids speak, thingy. Tracing the perimeter and then looking at the area and look at how much stellation, which is the wrong word, but how much they differ from circle. It looks like there's a lot in periphery, but you take out connection 43, you can kiss that goodbye, everything goes into body. And then people say that's irrelevant. Is it? If you look at Erwin Nair work, and you go back, they're shown in DRG neurons, there is a body release. And that body release gives you some information that those processes that are far away cannot tell you whatsoever. So I think that's a good question. All right, thank you guys. Sorry for there was a little time we
Now we have coffee break. Oh, I have two announcements. One important announcement is for students that got fellowships. We will send, after you go back to your own countries and your places, provinces, etc., you will receive an email from us with the form that you should complete with your bank account data, and we will transfer another form that is going to be for viáticos, so for traveling. To that, together with your boarding passes, everything will go back to us, and we will send, no, not us, the university will send you the money to your bank account. So everything is going to be after you return. But keep the boarding passes and keep posted waiting our email. If you are not getting our email in a week, just send us an email because something is wrong. Okay? Yeah. And now we have coffee break.
Yes,
We can back from the coffee break 10 minutes before 11, okay? 10 minutes
Yeah,
before.
we have 15 more minutes for the coffee
10
break.
minutes, you are.
Yeah, but it was, I was 5 minutes late. I was 5 minutes late. Thank you. Just cut it down and give you delete. You don't mind deleting.
You know what?
Yes.